Hello everyone and welcome back once again to the biology class of Fun Scholar. So today we are going to continue the chapter and finally wrap it up with its second part. So the chapter that we were dealing with was the flower, which is the second chapter of 6th grade in ICSE board. So now let's continue the discussion from where we had left in the last lecture. So as you can see in the last lecture we had learned about fertilization. So what is fertilization? So for recapitulation we can discuss that fertilization is the process of fusion of male and female gametes. So and it is seen in case of sexual reproduction, because only in case of sexual reproduction the male and the female gametes are produced and then they fuse together to form the zygote. And in case of plants also we can see such type of sexual reproduction where the male gamete is represented by the pollens produced by the anther and the female gamete is represented by ovum, which is or the ovule, which is present inside the ovary in the pistil. So therefore the transfer of pollen we have seen in the previous lecture from the anther to the filament or to the stigma of the flower, where they germinate and produce the pollen tube to reach the ovary and fuse with the ovule inside it. So that is known as pollination. So we have discussed till pollination and then the subsequent process of fertilization. So now we have known that after fertilization what will happen to the structure of the flower. So till fertilization we had done in the last lecture. Now today we will be learning about the structures produced post fertilization and also the we can say the categories of that structure. For example, fruit is produced after fertilization, so we'll be dealing with the types and the structure of the fruit. Then seed is produced after fertilization, so then we'll also study the structure of the seed, and also we'll know about the conditions required about the proper seed germination. And so these are the topics that we'll be dealing in today's class. So first change that occurs post fertilization is that the ovary enlarges. The ovary enlarges and develops into a fruit, while the ovules inside the ovary turn into seeds. So therefore, and the other parts which were protective in function, so like the sepals, the petals, the stamens, they wither and fall off because they are no more required and their function is over. So therefore they dry off and they fall. But the major changes are these two things, that the ovary turns over into the fruit that we can see, and the ovules turn into the seeds inside the fruit, which bear the zygote, and that zygote develops into the embryo that can further grow into a new sapling when planted in the right condition. Okay, so now let's proceed. Now, coming to false fruits. As I told you, in this lecture, we'll be knowing about the types of fruits also. So, false fruit is a type of fruit which is not produced just like the other fruits. For example, other fruits, that means from the ovary. So, in case just now we have learned that fruit is formed from the swollen and the ripened ovary. But in case of the false fruit, it is formed from other parts rather than the ovary, like the thalamus. We have already known in parts of flower that thalamus is the portion that provides the base of the flower. So when that thalamus swells up and develops into the fruit, that is known as false fruit. Example of some of such false fruits can say the apple is there, then the pear, then the grapes, then the banana. These are all the kinds and the types of the false fruits. Now, coming to the parts of a fruit. Now, fruit has two broad parts. We can say the pericarp and the seed. So, therefore, seed is present inside. It is the hard structure which bears the embryo of the new plant. So, that is the hard structure, and also the outer layer of the seed that actually compose the fruit, that is known as pericarp. So basically we can divide the fruit into two parts, the pericarp, that means the fruit wall, and the seeds. Now pericarp can be further divided into epicarp, mesocarp and endocarp. So you can see these things, that pericarp develops from the wall of the ovary. It can be thick or thin depending on the type of the fruit. So we can see so many varieties of fruits around us. So depending on the variety of the fruit, the epicarp structure can vary. So the epicarp structure, the pericarp, which consists of the epicarp or the exocarp, then the mesocarp and the endocarp. So exocarp you can say, or the epicarp, whatever it is the peel of the fruit. The peel of the fruit which we actually remove and then eat the fruit. So that peel is the exocarp or the epicarp. And what is mesocarp? Mesocarp is the, meso means middle. So mesocarp is the middle fleshy layer which is edible in nature. See, the sweet and fleshy edible middle layer. This is the mesocarp. Now, it is not always fleshy. It can also be hard depending again on the type of the fruit. But the middle portion which is edible, that is known as the mesocarp. And what is endocarp? The innermost layer. The innermost layer is the layer which will enclose the seed. So therefore it can provide protection to the seed structure also. So that is known as the endocarp. It is clearly visible in case of, for example, mango. You will see the outer layer or the peel of the mango that represents the epicarp or the exocarp. Then the juicy fleshy middle layer represents the mesocarp, and finally there is a hard inner layer which protects the seed of the mango, that represents the endocarp. So therefore these are the three layers of the fruit, and these three layers together, they are known as pericarp, right? And inside the pericarp lies the seed which has developed from the ovule after fertilization. Now again, coming to the types of fruits, as I told you earlier also, fleshy fruit and dry fruit. So obviously from the name only you can understand that fleshy fruit will be those type of fruits in which the pericarp will be soft and edible, right? Just like mango, tomato, then the plum, cherry, where the fleshy means which is very soft and full of flesh and juicy. So therefore this, the wherever we will find that the pericarp is juicy, fleshy and soft, then that type of fruits are known as fleshy fruits. And dry fruits, we already know that includes the nuts. So the pulp is hard. That is also edible, but that is not soft. The pulp is hard. So therefore it is known as dry fruit. And it also contains the seeds. For example, the pea pod, in the pea pod also is a type of dry fruit which contains the seeds, that means the peas. Then walnut shell. So within that walnut shell we have the seeds of the walnut which can germinate into the new walnut tree. Now coming to the functions of a fruit. So do fruits have any particular function or any significant function? Yes, of course. Since apart from being the edible part, so it also has some particular roles to play in the structure of the plant. First of all, it protects seeds from unfavorable conditions. Yes, of course, because seeds are contained within the structure of the fruit. So therefore, fruit will be providing protection to the seeds. Then it will store the food inside. So fruit acts as the storehouse of the food. So we know that leaves are known as the kitchen of the plant, and whatever excess food that is not utilized and immediately by the plant, that is stored in various organs such as the stems, the roots, and apart from them, fruit is also another structure in which food is stored. So therefore it acts as the storage organ of food for the plant, and also it aids in dispersing seeds and facilitating germination. We all know that we love to eat fruits, be it the fleshy fruit or the dry fruit, whatever it is. So whenever we eat the fruits and then we throw away the seeds here and there, so in doing that, the seeds are dispersed from one place to another. So in this way, the fruit is actually helping in the seed dispersal also, and if the seeds fall on certain on substratum on which it can grow properly because it has got all the other amenities, so then it can germinate to form the new sapling. So these are the three important functions that a fruit performs for the plant. Now coming to the seed. Now after fruit, we need to know about the structure of the seed and also the types of the seed. So what is seed? Seed is the fertilized ovule. When the ovule is fertilized by the male gamete, so then it turns into the seed and it is protected, just now we have discussed, inside the fruit usually. Now it is a hard structure usually, and it contains the embryo, which is the future plant sapling. Now what are the types of seeds? Firstly, there are two types of seeds. One is the dicotyledonous seeds and monocotyledonous seeds. So what do we mean by these two types? Let's see. Now mono means one. We have already discussed this once in the chapter the leaf, but this is for the recapitulation also. And cotyledon means seed leaf. Right. So therefore, the seeds which contain only one seed leaf, those type of seeds are known as monocotyledonous seeds. Now what is seed leaf? Seed leaf actually are the structures which are present inside the seeds which contain the food reserve for the growing embryo. It actually sustains the life of the embryo of the new plant. The new plant remains in the embryo stage in the arrested form until and unless it gets the proper environment to grow, for the seed to germinate and grow. So but it is alive, so in order to keep it alive or to sustain its life, it needs nutrition. Now who will provide this nutrition? This nutrition is provided by the seed leaves. So the seed leaves actually act as the storehouse of nutrition for the embryo inside it. So therefore, the seeds which have got only one seed leaf, those are known as monocotyledonous seeds. For example, wheat, maize, rice, all those things. And then similarly dicot. Di means two, right? And again cotyledonous means seed leaves. So the seeds which contain two seed leaves or which contain food reserve in two seed leaves, those type of seeds are known as dicotyledonous seeds. Example, beans, peas, they are there. So these, you can see the structure also, the bean seed structure, which is the example of dicot seed, and the corn seed structure, which is the example of monocot seed. So here you can understand this, that if you break open the dicot bean seed, what you will get to see, you will see the outer layer of the seed which is known as the seed coat, right? Just like coat is the covering that we put on during the winter to protect ourselves against the cold. So similarly, this seed coat protects the seed from the environmental conditions which can be adverse. So this, then embryonic leaves means the leaves which are present in the embryo stage, right? Then embryonic root also is there, which will further develop when planted in the soil. And then cotyledons, the two seed leaves which contain the food reserve for this embryonic stage of the new sapling. Then coming to the monocot seed, that is that corn, and then that is the cotyledon, you can see here. So on one side only you can see the food reserve because it is a monocot type of seed. So then again it will contain the embryonic leaf, then the fruit tissue which will be present surrounding the seed, then the seed coat, endosperm is there, then embryonic shoot, embryonic root, and the sheath. So a layer of protection which is represented by the sheath. So the again see, these are the examples. So based on the number of cotyledons, seeds are of two types, the monocot seeds and the dicot seeds. So therefore again here you can see the first true leaves of the monocotyledonous corn seeds. See, the leaves are there, single cotyledon is there, and here in this case two cotyledons are there, right? So it is best represented if you take these type of seeds and then break open and try to understand this. Now, monocot seeds such as the maize or the corn has got one cotyledon, and here the endosperm stores food in the form of starch. So we know that in case of plants, the stored form of food is starch only, because plants produce glucose as a result of photosynthesis, and that glucose is stored in the form of starch in the storage organs. And among the storage organs, there is another storage organ, that is the endosperm. What is this endosperm? Endosperm actually is formed when after the fertilization process only, when the part of the pollen grain or the male gamete fuses with the polar nuclei, that you will be studying in detail in higher classes. So then it forms the endosperm. It is nothing but just like the food reserve only, that will provide nutrition to the growing embryo, and it is surrounded by a protein rich aleuron layer. So therefore the aleuron layer is there which will provide proteins. Now why are proteins required? We all know protein is bodybuilding in nature. So in this manner it will provide the protein to the growing embryo so that the embryo can develop properly. Then the cotyledon contains the embryo, the radicle, and the plumule. What is this radicle and the plumule? The radicle is the portion from which the root will grow, and the plumule is the portion from which the shoot will grow. So these are the two parts which contain, and in the embryonic stage they are known by these two names. Now coming to the detailed description of dicot seed such as the bean seed, it is protected by a thin outer coat called the seed coat that I have already discussed, and it consists of two parts. So the testa and the tegmen. What is that? Testa is the outer protective layer. You can see that which is present beneath the seed coat. That layer is the testa and the tegmen. So it's in the seed coat only, and tegmen, it is the inner thin layer. The cotyledon store food for the growth. The embryo consists again the radicle which develop into the root, and the plumule will develop into the shoot. So therefore, now what is this micropyle? Micropyle is nothing but a pore through which the nutrients and the minerals will enter the seed, and then it will be absorbed for the growing embryo. for its development. So the pore is known as the micropyle. It is present in the ovary also, where through which the pollen tube enters the ovary and then it carries the male gametes to fuse with the female gamete, that is the ovule. Now that micropyle is retained when it gets converted into the seed also. See, this is a comparative table between the dicot seed, that is the bean seed, and the monocot seed, that is the maize seed. Now, in case of the dicot seed, the endosperm is absent. Now, you can say why? Because in case of the dicot seed, it contains the two seed leaves. So, it has got enough nutrition for the growing embryo. But since in case of the monocot seed, the endosperm needs to be present to provide the extra nutrition which cannot be fulfilled by the single seed leaf. So therefore, the endosperm is present to provide the nutrition. Now that structure is actually giving the extra support. Now cotyledon store food, here is the endosperm here stores food, and cotyledon is also there. We know that a single cotyledon is there. So inside that single cotyledon, this endospermic structure which is there, this is the one that stores food. Embryo is large and embryo is small in this case. Seed is contained separately in a fruit. Seed coat and fruit wall are fused to form a grain. So therefore, naturally, whenever we see the grains, so they are all the representatives of monocot seeds, because in case of grains only we can find that the seed coat and the fruit wall will fuse to form a small hard structure. But in case of this seed, that is the dicot type of seed, it is present separately, and therefore the layers of the fruit that we have studied, that is the epicarp, mesocarp, and then the endocarp, and that of the seed, that means the seed coat, they remain separately. Now coming to the next topic, that is seed germination. What is seed germination? This is a very common topic. So seed germination is the process by which the embryo within the seed becomes active. As I told you, it remains in an arrested form until and unless it gets the favorable conditions to grow. So that becomes active and grows into a young plant in the presence of water, air, and suitable temperature. So unless and until it gets the proper substratum to grow, either the soil or the, for example, we know that there are experiments in which we can grow sprouts within cotton sheets also. So that also acts as the base or the substratum. So unless and until it gets the proper substratum to grow, and along with that the proper conditions, that means the adequate amount of water to perform the process of photosynthesis, and also we know water is an essential part of all the living organisms, because the living organisms cannot survive without water. Then we have got air, from which it will get the carbon dioxide and oxygen for photosynthesis and respiration respectively, and the suitable temperature. And this suitable temperature varies from one seed type to another, because it depends. In higher classes you will be learning about the different types of crops. So some crops require humid type of temperature, some require dry temperature, some requires the type of temperature which is very cold temperature. So therefore it depends on the species of the crop. So therefore the suitable temperature for that particular type of seed, that is required for that seed to grow. And if any one of this factor is missing, then obviously the seed will not grow properly and it will not give rise to the growing plant. So germination, you can see here that unlocking life's potential. Why is it unlocking life's potential? Because life is contained within this embryo when it is living, right? And it has got the potential to grow into a new plant or a new tree and then continue the cycle of life and give rise to the next generation of offsprings. So that is actually done by the embryo which is contained within the seed. Now, first, these are the steps by which germination occurs. The water absorption. The seed will first absorb the water and then it will swell up, right? It will make the embryo which was present in arrested form inside the seed active, right? And then the radicle, that means the root portion, will emerge first, and that is known as the sprout. That is known as sprouting, and the radicle which comes out first, which is usually white or whitish in color, so that is known as the sprout. And then we say that that particular seed is sprouting. And we also can take the sprout. It is very rich in nutrients because it is rich in the proteins we have already seen, and also the seed leaves which are rich in nutrients which provide nutrition for the growth of this sprout. They are usually active at that stage. So therefore, if we can intake this sprout, it is very healthy for us as well. Then plumule emerges. Then after the radicle, the plumule or the young shoot will appear. The germination turns a seed into a seedling. That means when the radicle and the plumule will come out of the seed, then we call that a seedling. Now, water, oxygen and temperature are vital. So therefore, after that, after the seedling has come out, then it needs to be planted properly in the soil, or if it is planted already in the soil, then also no issues. It needs to be taken care of properly in the initial days because it is very delicate when it is growing. So in the seedling stage, then the sapling stage, it is very delicate unless and until it becomes accustomed to its environment, the physical environment in which it is growing. Then these are the steps. We can say that first, water absorption, the seed will absorb the water, that will initiate the process of germination. Then the seed will swell up as it will take up more water and it will prepare for the growth of the embryo. It will make the embryo active. The nutrients which are there, that will also be actually passing on to the growing embryo so that it can develop quickly to the seedling and then consequently into the sapling. Then breaking open, the seed pod splits allowing the embryo to emerge. So as the seed gives rise to the seedling, we can say that slowly and steadily the seed pod, then the seed leaves, their importance slowly starts deteriorating because there is, as the seedling becomes independent enough to take up its own nutrition. So it is that point in which the importance of the seed pod, then the seed leaves, they start to decrease, right? And then finally the growth of the embryo, in which the embryo grows into a seedling, completing the germination process. So germination process starts with the absorption of water and ends with the growth of the embryo into the seedling. So from water absorption to the growth of the seedling is the span which we call the seed germination process. Now after this seedling, then also further growth continues until and unless it develops into a mature plant or tree. Okay, but germination process is actually recorded till the process where the seedling will come out of the seed. Now there are again two types of germination specifically. One is the apigeal germination, another is the hypogeal germination. Now what is apigeal? The term api means exterior or above, right? Just like we have read about epicarp means the exterior covering. So epi means exterior or above. So it is a type of germination in which the cotyledons are pushed above the ground, right? You, for example, the bean seed is there, so you can see here, after the growth of the sprout or the root, then as the root grows, you enlarge the root height that increases, then the cotyledons, they are pushed out. They actually shed the seed coat, and then as I told you, their role is now over. So they will shed the seed coat and then they will be pushed out of the soil or the level of the ground in which they are planted. So now then it will grow further into the seedling when the plumule will also grow out. So, and then you can see the cotyledons will be present in the vestigial form, and slowly they will also wear out and then fall. So this is the process of germination which takes place above the ground. That is why it is known as apigeal germination. So the leaves unfold and start preparing food. So therefore, once the plumule develops into the shoot which contains the leaves, so then the leaves will start preparing the food by the process of photosynthesis, and then when it can prepare its own food, then there is no more use of these cotyledons and then they will fall off. And what will happen in case of hypogeal germination? Hypo means below. Below what? Below the level of the soil of the ground in which the seed is sown, right? So in this case, the cotyledons or the seed leaves remain below the ground only. Example, the peas. So what happens, you cannot see this is the case of hypogeal germination in which you cannot see the cotyledons above the surface of the ground. You can only see the epicotyle or the plumule which has grown into the shoot, the young shoot, which is known as epicotyle, right? So that is the portion that you can see above the ground, and you cannot see any other structure apart from it. The root structure will remain under the soil only. That is always very evident because they absorb the water and minerals from the soil, but the cotyledons also remain below the level of the soil. And similarly, when the plant is able to prepare its own food with the help of photosynthesis, then the cotyledons will wear out and will slowly dry off in that, but that will happen inside the soil only. That also you cannot see from outside, that is not visible. So this is the case of hypogeal germination, in which all the development and then wearing out of the cotyledons will occur inside the soil or below the level of the ground. And on the other hand, in apigeal germination, the cotyledons are pushed above the ground as the radicle comes out and develops into the root, and then the cotyledons, they actually wear out and fall once the leaves start preparing their own food. So these are the two major distinctions. So that was all regarding this chapter. I hope you have now understood the basic concept regarding the structure of the flower, how pollination and fertilization takes place, what are the events which are mandatory or prerequisites for fertilization to take place successfully, and also what are the events that take place post fertilization, then about the structure of the fruits, then the seeds, and also the types of the fruits and the seeds, and also of germination that happens post the fertilization. So if you have got any queries regarding all these topics, so you can again post your queries in the comments below, and again we'll meet in the next lecture with another chapter. So till then, stay tuned, take care, thank you, bye bye.